Welcome to the new era of video creation with BioZ8. Today we will discuss with AlphaFold. Here are the contents. AlphaFold, developed by DeepMind, is a state-of-the-art deep learning system designed to predict the 3D structures of proteins based solely on their amino acid sequences. This is a landmark achievement in the field of structural biology. The protein folding problem has eluded scientists for decades. Predicting how a chain of amino acids folds into its final 3D form has been a challenge because the number of possible configurations grows exponentially. In 2018, AlphaFold made headlines when it won the CASP-13 Critical Assessment of Structure Prediction competition. Then, in 2020, it dramatically improved its results in CASP-14, essentially solving a problem that has stumped researchers for over 50 years. The system's success holds massive implications for drug design, bioengineering, and our overall understanding of biology. Proteins are the molecular machines of life, responsible for nearly every biological process. These molecules are made up of chains of amino acids, which fold into complex three-dimensional shapes. The unique shape of each protein is critical because it directly determines its function, whether it's catalyzing reactions, transmitting signals, or forming structures within cells. The central problem, however, is that predicting how a protein will fold into its 3D shape based only on its amino acid sequence has been extraordinarily difficult. The number of possible configurations a protein could adopt increases dramatically with its size, making exhaustive computational search infeasible. Despite the significance of this problem, no reliable computational solution existed until AlphaFold came into play. AlphaFold utilizes deep learning, an approach inspired by the structure and function of the human brain, to solve the protein folding problem. One key aspect of AlphaFold is its use of transformer-based architecture, a model originally developed for natural language processing tasks, but adapted for protein structure prediction. The model is trained on large datasets of known protein structures, learning the relationships between amino acid sequences and their 3D structures. Unlike traditional methods, which relied heavily on simulating physics-based interactions, AlphaFold also integrates a wealth of biological and physical knowledge to guide its predictions. It specifically focuses on predicting the interactions between amino acid pairs, which play a significant role in how the overall protein folds. Protein folding involves more than just local interactions between adjacent amino acids. One of the challenges is that folding is a highly complex process with many intermediate states, some of which are not stable. Proteins also have long-range interactions between amino acids that are far apart in the linear sequence, making the problem even more difficult to model. Another challenge is the scarcity of experimental data, as experimentally determining protein structures is time-consuming and expensive. Most of the data AlphaFold uses comes from publicly available databases like the Protein Data Bank. Additionally, predicting protein structures accurately involves heavy computational power. Prior to AlphaFold, simulations of protein folding took days or weeks to run, making it impractical for large-scale predictions. AlphaFold's core algorithm begins with an input sequence of amino acids, representing the primary structure of the protein. The model then predicts the 3D coordinates of each atom in the protein. To achieve this, AlphaFold uses a neural network that has been trained on evolutionary data, e.g., homologous proteins, spatial constraints, and physics-based knowledge, such as bond angles and distances. The model makes use of attention mechanisms, a technique from the transformer architecture, to focus on the relevant amino acid interactions. These attention mechanisms allow the model to weigh the importance of different parts of the protein sequence and their relationships, which is essential for predicting how distant amino acids interact within the folded structure. Evolutionary data plays a crucial role in AlphaFold's performance. Proteins evolve over time, and through this process, certain amino acids in a protein are conserved because they are critical to the protein's function or structure. By studying multiple sequence alignments, MSAs, collections of related protein sequences, AlphaFold can identify conserved patterns that suggest important structural features. These evolutionary signals are especially useful for predicting interactions between amino acids that are far apart in the sequence but close in the folded structure. By learning from homologous proteins, proteins that share a common ancestor, AlphaFold can refine its predictions and make them more accurate, even in the absence of experimental data for a particular protein. Prior to AlphaFold, protein structure prediction typically relied on methods like homology modeling, which builds models based on the known structures of similar proteins. These methods have limitations when a close homologue is not available. AlphaFold changes the landscape by providing predictions that rival experimental accuracy, even for proteins with no homologues in the databases. This breakthrough significantly reduces the reliance on time-consuming and costly experimental techniques, such as X-ray crystallography and NMR spectroscopy. With AlphaFold's predictions, scientists can now explore the structures of proteins that were previously inaccessible, which has immense potential for accelerating drug discovery, vaccine development, and the design of synthetic proteins. The Caspi competition is one of the most important benchmarks in protein structure prediction. In CASP 13, 2018, AlphaFold emerged as a leader, significantly outperforming other methods. However, it was during CASP 14, 2020, that AlphaFold's performance truly blew the scientific community away. AlphaFold achieved near-experimental accuracy for nearly every protein tested, including those that had long been considered too complex for reliable predictions. By comparison, traditional methods, even those based on years of research, could not match AlphaFold's accuracy. This achievement highlighted AlphaFold's transformative capabilities and marked a new era in computational biology, where AI can directly assist in solving biological problems. The potential applications of AlphaFold are vast and span multiple fields. In structural biology, AlphaFold accelerates research by providing highly accurate 3D models of proteins, reducing the need for expensive and time-consuming experimental methods. In drug design, understanding the precise 3D shape of a protein allows researchers to design small molecules that can specifically target and interact with the protein. Protein engineering can also benefit from AlphaFold's predictions, enabling the design of synthetic proteins with desired properties. In disease research, many diseases are associated with misfolded proteins, such as Alzheimer's and Parkinson's. AlphaFold can help researchers investigate these misfolded structures, potentially leading to novel treatments. One of the most immediate and impactful uses of AlphaFold is in drug discovery. In the past, discovering drug targets and designing molecules to interact with them involved a lot of trial and error, often requiring expensive and time-consuming experiments. With AlphaFold, researchers can quickly generate accurate 3D models of proteins that are drug targets, enabling faster identification of potential drug candidates. Moreover, AlphaFold can help predict protein-ligand interactions more accurately, potentially speeding up the process of drug design. AlphaFold can even facilitate the study of personalized medicine by modeling how specific proteins in individual patients may behave differently, leading to more effective and tailored treatments. While AlphaFold represents a major breakthrough, it does have some limitations. One key limitation is that AlphaFold primarily predicts static structures, meaning it does not account for the dynamic nature of proteins. Many proteins undergo conformational changes as part of their function, and AlphaFold currently cannot model these transitions. Additionally, AlphaFold has some difficulties with membrane proteins, which are embedded in cell membranes and have unique structural characteristics. Protein complexes, groups of proteins that interact with each other, are also more challenging for AlphaFold to model accurately. Finally, the model's accuracy is still dependent on the quality of the training data, and areas with limited experimental data may lead to less reliable predictions. As remarkable as AlphaFold's achievements are, there is still room for improvement. Future iterations of AlphaFold could focus on improving the model's ability to handle dynamic proteins that undergo conformational changes. Further advancements may allow AlphaFold to predict interactions between multiple proteins in a complex. Another key area of focus is membrane proteins, which remain a challenge due to their unique structural and environmental characteristics. Additionally, integrating AlphaFold with other AI-driven biological models could create a more holistic understanding of biology. Beyond drug discovery, AlphaFold's capabilities may also extend into synthetic biology, where it could be used to design novel biological systems for manufacturing and other purposes. One of the most notable aspects of AlphaFold is DeepMind's commitment to open science. In July 2021, DeepMind released AlphaFold's source code and the predicted structures of over 200 million proteins in a publicly accessible database. This massive dataset includes proteins from across the entire tree of life and is available to researchers worldwide, fostering collaboration and accelerating research in numerous fields. By sharing this data, DeepMind has not only democratized access to cutting-edge technology, but also encouraged transparency and open collaboration in structural biology, paving the way for more discoveries and innovations. AlphaFold is already making significant contributions to structural biology and has the potential to revolutionize this field. By providing highly accurate predictions of protein structures, it bridges the gap between computational biology and experimental methods. This accelerates the pace of research in areas such as disease mechanisms, drug discovery, and bioengineering. As AlphaFold's predictions continue to improve, it will support new breakthroughs in understanding the molecular basis of diseases and in developing new therapies. The broader impact will be felt across many disciplines, from biochemistry to genetics, as AlphaFold unlocks new possibilities for scientific exploration. In conclusion, AlphaFold represents one of the most significant breakthroughs in both artificial intelligence and structural biology. Its ability to predict protein structures with near-experimental accuracy is transforming the way we approach a wide range of biological challenges, from drug discovery to synthetic biology. By releasing its source code and protein predictions publicly, DeepMind has made this groundbreaking technology accessible to the global research community, accelerating scientific progress across disciplines. As AlphaFold continues to evolve, its impact will be felt for years to come, shaping the future of biology, medicine, and AI.